Welcome dear students. In this video we are going to learn about a very important cell signaling pathway that is called mTOR pathway. This pathway actually controls cell proliferation, cell growth and cell survival. But at first I will give you some basic concept about this pathway and then I will go into details. The first thing you have to understand why it is called mTOR pathway. The name mTOR comes from mammalian target of rapamycin so mammalian target of rapamycin. Okay, so a molecule called rapamycin that can target a protein that is present in mammalian cell. So the rapamycin is actually a chemical that is produced by a strain of bacteria called Streptomyces hygroscopicus. And this strain was discovered by a Canadian expedition in 1964 in the Easter Island. And in 1972, an Indian scientist, Dr. Surendranath Sehgal, discovered this chemical from this bacterial strain. It is also known as sirolimus. This rapamycin, at first it was found that this has an antifungal activity, but subsequent research found that this molecule can prevent transplantation rejections, specifically in kidney transplantations. Today also this molecule is used. It is actually a FDA approved medicine. This is known as rapamycin or sirolimus. This molecule can also prevent cancer. Now this molecule, this is the rapamycin. At first it was found that in yeast it can prevent a protein that called TOR, target of rapamycin. And eventually it was found that this rapamycin can also target another protein that is present in mammalian cells called mammalian target of rapamycin. So this is the naming. I will go into more details about this signaling pathway. At first I will show you the initiation of this molecule, how this pathway actually activated. Then I will show you the function of this pathway. The first thing I have mentioned in my previous class that both the JAK and STAT pathway, both this pathway can activate 4 signaling pathway. Both this pathway can activate STAT protein. Both this pathway can activate GRB2, a adapter protein through which RAS-MAP kinase pathway can be activated. Because I have mentioned that in case of JAK-STAT pathway or RTK pathway, the receptor for the ligand actually gets phosphorylated at the tyrosine residue. The name receptor tyrosine kinase, that means the receptor gets phosphorylated at the tyrosine amino acid. And in case of JAK-STAT pathway also, the receptor gets phosphorylated at the tyrosine residue. And the tyrosine residue, when gets phosphorylated, it can bind with a domain of protein called SH2 domain. SH2 domain binds with phosphotyrosine. You have to understand the basic fact about this, then you can understand the whole pathway. This is a long story. SH2 protein, SH2 domain, actually a domain, can bind with phosphotyrosine. So those proteins have SH2 domain can bind with this receptor. Like GRB2 is an adapter protein, have a SH2 domain. So it can bind with these 2 receptor. Then PLC gamma, it also has SH2 domain. And we know that PLC gamma activate IP3 DAC or produce IP3 and DAC that can activate PKC, and also calcium concentration increases. Another thing that is Pi3 kinase. This enzyme actually associate with the RTK or JAK-STAT pathway because Pi3 kinase also has SH2 domain. And then this Pi3 kinase activate a kinase also called PKB, protein kinase B, or it is also known as AKT. Then they actually activate and perform the functions. This is actually kinase. I will show you how this actually occurs because they can activate the mTOR pathway. This protein can activate the mTOR pathway. So today's class actually will focus on this molecule, how this molecule actually gets activated and actually subsequently activate the mTOR pathway. Okay, I will not tell you about this molecule, only this molecule, this Pi3 kinase. So what happens? I have mentioned in the previous classes always that this is the plasma membrane and there is a receptor. Let's suppose this is the receptor These are the two receptors when any growth factors, because RTK pathway can be activated. If you have not watched my video, RTK pathway, must watch this video at first. RTK JAK-STAT I have mentioned. I have tick mark here that I up to RasMap kinase pathway I have take the separate class. Now I am talking about mTOR pathway. When any growth factor binds, or any cytokines, that could be, because JAK-STAT pathway can be activated by cytokines or hormones like growth hormone, prolactin, thrombopoietin and erythropoietin. And the RTK pathway can be activated by growth factor, insulin like growth factor, fibroblast growth factor, EGF, epidermal growth factor, any growth factor. So let's suppose this is the growth factor. Okay, when growth factor binds, the receptor actually dimerizes, and ultimately the receptor gets phosphorylated at the tyrosine positions. And then different proteins, I have mentioned here, different proteins can bind, and they can actually spread the message through different cell signaling process. Now what happens when this protein or this receptor gets phosphorylated? This Pi3K can bind. Let's suppose this is the Pi3K. Pi3K, the name implies phosphoinositide 3 kinase. The name means phosphoinositide. Pi3K means phosphoinositide 3 kinase. We know that in the plasma membrane there is a phosphoinositide we call phosphatidylinositol, Pi. This is a phosphoglyceride. This phosphatidylinositol has inositol sugar. In that sugar, the sugar can be modified by phosphorylation. Here the kinase phosphorylates at the carbon 3 position of the inositol, so it is called Pi3 kinase. Okay, the Pi that means phosphatidylinositol can be into one form actually called Pi4 phosphate, and we know that this phosphatidylinositol is actually mainly concentrated in the cytosolic leaflet, not the extracellular leaflet. This is the cytosolic leaflet. The plasma membrane is bilayered in structure. This is the cytosolic leaflet and this is the exterior leaflet. The Pi is concentrated in the cytosolic leaflet. Now this Pi45, this is already actually this Pi means phosphatidylinositol is already added with phosphate at the position of 4. Carbon 4 is phosphorylated initially. Then Pi3K can phosphorylate this Pi345 phosphate and this become Pi34 bisphosphate, or I can say P2. Okay, at first it was 4, but when Pi3K phosphorylated it will become Pi34 P2. Another kinase is present called Pi5K. That means it will phosphorylate at the carbon 5 position of the inositol, because here the inositol is a sugar. Inositol is a sugar. This sugar can be phosphorylated at the carbon 5 positions. So when this Pi45 gets phosphorylated at the 5 position, it will become, by this enzyme, it will become Pi45P2. And we know that this PLC gamma or PLC beta actually phosphorylate, actually degrade or hydrolyze this Pi45P2 to IP3 and DAC to make this IP3 DAC. Now this Pi45P2 can also be phosphorylated by this Pi3K and then it will become Pi345P3. You can see 3 position gets phosphorylated. And this 2 inositide is the docking site. You have to understand the pathway. I will go to the mTOR, but I will show you the first initiation. This Pi3K, at first growth factor binds, this receptor gets phosphorylated at the tyrosine position, and then Pi3K bind with the SH2 domain, and Pi3K phosphorylate the phosphoinositol, that is a phosphoglyceride present in the plasma membrane, and then it actually becomes a docking site. The same is for the tyrosine. I have mentioned that SH2 means phosphotyrosine binding domain. Those proteins have SH2 domain can bind with phosphotyrosine. The same is true. Those proteins have pH domain. pH domain means plextrin homology. Those proteins have pH domain can bind with this phosphoinositide molecule. One example of this protein that have the pH domain is the PKB or AKT. You must remember the two name also. This PKB or AKT is actually a serine threonine kinase. It can phosphorylate at particularly serine and threonine, not in the tyrosine kinase pathway. But when there is no growth factor, this PKB and AKT is inactive in conformations. This PKB and AKT is off when there is no growth factor. But when growth factor binds, then this Pi3K phosphorylate these two, like Pi45 to Pi34P2 and Pi45 to Pi345P2. Then this PKB, which have the pH domain, bind with these two molecules. The pH domain present in this PKB, prevents its function. When pH domain bind with this molecule, then the PKB actually starts to get activated. But not only the binding of pH to these two molecules will not activate the PKB. Another protein that has the pH domain through which it can bind with these two molecules are called PDK1, phosphoinositide dependent kinase 1. This PDK1 is a kinase. This PDK1 also has pH domain through which it can bind with these two molecules, and by this way this PKB kinase and PDK kinase come very close to each other. You have to understand this. When there is no growth factor, then there is no docking site, because then 3-phosphate 3,4 and 3,4,5 is not present. Then these two molecules remain inactive. But when this growth factor binds, this Pi3K gets activated and they phosphorylate and actually make this docking site. In that situation, PKB and PDK, those have pH domain, bind with these two molecule and come close to each other. This PKB, as it is a kinase, so it has an activation loop. PDK1 phosphorylate at the threonine at the threonine position of the activation loop, and also a molecule called PDK2 also phosphorylate at the serine position of this PKB molecule. Okay, so these two kinase helps in the activation of this PKB or AKT. Both this PDK1 and PDK2 helps in the activation of PKB or AKT. Yes, another molecule that can actually helps in the activation of PKB is called mTOR protein complex 2. Now you will get confused because I have mentioned that PKB or AKT pathway can activate mTOR pathway. Now I am telling you that mTOR C2 also helps in the activation of PKB. How it can occur, I will tell you in the last of this class. At first you learn about this. mTOR C2 is a complex. This mTOR protein can be two form. This mTOR protein can be two form. One is called mTOR 1 C1, that means complex 1, and another is called mTOR C2, complex 2. In the complex 1, there is a protein that associates with this mTOR protein called Raptor. In the complex 2, there is another protein called Rictor. When mTOR is associated with Rictor, it's called mTOR C2. This mTOR C2 helps in the activation of PKB because mTOR C2 changes the conformation of the PKB so that the PDK1 can phosphorylate at the threonine position of this activation loop and it becomes activated. And by the way, PDK2 also phosphorylate at the serine position, and when both these kinase phosphorylate this PKB, the maximum activity of PKB actually comes. Then it can function maximally. Okay. Now this PKB or AKT function in three ways, actually perform three important functions. The first thing you have to understand is, I have mentioned that this mTOR pathway actually perform or control three things. One is cell growth, cell proliferation and cell survival. The first thing that is performed by PKB or AKT, this is actually a kinase, serine threonine kinase, that is a pro-apoptotic protein called BAD. This is called BAD, pro-apoptotic protein. That protein helps in the apoptosis. This is called pro-apoptotic protein. This protein helps in the apoptosis. Then the cell will not survive, it will die. This protein called pro-apoptotic protein BAD actually bind with another protein called BCL2. This is a family of anti-apoptotic protein. This is an anti-apoptotic protein. You have to understand this. Pro-apoptotic protein binds with anti-apoptotic protein and prevents its functions. But when growth factor binds, when this chain reaction occurs, when PKB gets activated, this PKB phosphorylate the BAD at serine or threonine position, because it is a serine threonine kinase. When BAD gets phosphorylated, it no longer binds with BCL2, and BCL2 actually removed and actually become activated. Then BCL2 family proteins, that is anti-apoptotic protein, becomes actually active when it is not associated with BAD. And the BAD, when get phosphorylated, they also captured or sequestered by a protein called 1433. I have mentioned in my previous class. This protein actually gets captured by a protein called 1433. This 1433 acts as this domain, like SH2 domain binds with phosphotyrosine, pH domain binds with Pi3 phosphate, means phosphoinositide 3-phosphate. Where there is phosphoinositide 3-phosphate, pH domain, those proteins have pH domain can bind. The same is true for 1433. Those proteins have phosphoserine, they can bind with 1433, and 1433 is a cytosolic chaperone that actually prevent the function of the protein. Like when 1433 capture BAD, the BAD is inactive, it cannot actually bind with the BCL2. Then the BCL2 gets activated, so the anti-apoptotic protein helps in inhibition of the apoptosis. The first thing I have mentioned, cell survival. So by this way, PKB or AKT actually helps in the cell survival. Now, another way by which PKB works, that is called phosphorylation of FOXO3A, a fork head transcription factor. It is actually a transcription factor. So you can see here BAD gets phosphorylated and gets inactivated. Here also a transcription factor gets phosphorylated and also captured by the same protein called 1433. So what is the role of FOXO3A? Why it should be inactivated by this PKB? Because this FOXO3A binds with the DNA that promote transcription of this pro-apoptotic protein. Pro-apoptotic protein. You have to understand this. FOXO3 is a transcription factor. When there is no growth factor, it actually helps in the transcription of pro-apoptotic protein like BAD. So the cell can go into apoptosis. But when growth factor binds, when this chain reaction occurs, then the PKB phosphorylates the FOXO3A. Then FOXO3A can no longer present in the nucleus. It actually come into the cytosol and captured by this 1433 protein. Okay, by this way it actually prevents the cell survival, actually control the cell survival. So this is the two way. Another way I will mention here, that is another way PKB perform, that is the third way, control. That is, it can phosphorylate a protein called GSK3 beta, glycogen synthase kinase. Is also a kinase. The GSK3 beta phosphorylate and inhibit a protein called EIF2B. This is an initiation factor for translation. EIF means initiation factor, E for eukaryotic initiation factor 2B. When this protein gets phosphorylated by this GSK3 beta, it actually cannot perform its function, and the global translational initiation is stopped. That means you cannot activate the cell for translation when EIF4B gets inactivated. When EIF2B gets phosphorylated, no longer you can activate the translation, so global translation inhibition occur. So GSK3B inhibit this protein, and by the way, translation get initiation of translation stop. Now PKB or AKT perform as the inhibitor of GSK3B. AKT or PKB, I am writing the both, inhibit this inhibitor. When this that means it actually phosphorylate this protein, and when GSK3 gets phosphorylated, it cannot function. So you can see here the cell actually following a circuit. It can inhibit the inhibitor. When the inhibitor gets inhibited, then it cannot inhibit the target protein. So by this three way, PKB controls cell survival and translations. That means cell will proliferate. Now how this pathway, this Pi3 kinase, that means I am talking about this Pi3 kinase and PKB actually related with mTOR pathway. Yes, I will show you. The thing is here, when there is no growth factor, you have to understand thing. When there is no growth factor, and when there is growth factor, I have mentioned that growth factor can initiate RTK pathway, then RTK actually bind with the receptor, bind with Pi3 kinase, then the thing signaling occurs. But how this actually related with the mTOR pathway, I will tell you. The thing is, when there is no growth factor, the mTOR C1, that means this complex, not this complex, because I have mentioned this complex actually activate, this complex activate, helps in the activation of AKT or PKB, I have mentioned. Okay, this complex helps in the activation of PKB AKT, and this complex also activate a GTPase, or actually a RAS GTPase like protein called Rho GTPase. And we know that when Rho GTPase like protein, it is actually a RAS family protein, Rho GTPase protein, the example like Rho, RAC and CDC42, when gets activated, they control the cytoskeleton rearrangement. So the function of this mTOR C2 is very different, and I want to mention a very important information for your CSIR preparation, that rapamycin cannot inhibit this protein. It is insensitive and perform very differently. mTOR C2 means Richter complex. It is insensitive for by the prevention of rapamycin. Only this protein can be inhibited, mTOR C1, by rapamycin. I am only writing rapa. Can be prevented by rapamycin. Also by stress, hypoxia. Okay, this protein is actually sensor, but this protein is not very actually related with mTOR pathway, but this complex can activate the AKT or PKB, and I will show you how AKB PKT actually control the mTOR C1. So this is actually a very intricate circuit like structure. This protein can activate AKT and AKT can activate this mTOR C1. How? I will show you that. This mTOR C1 protein actually becomes activated by another protein called RHEB, REP GTP. This REP GTP also a family of RAS family, like Rho is a RAS family member, REP also a RAS family G protein, look like G protein because it associates with GTP. And I have mentioned that REP, when it is associated with GTP, it is on. The REP, same REP, when associated with GDP, it is off. So there should be, sorry, it is the REP. So there should be a GEF and GAP. GEF means those actually transfer this, and GAP means those actually convert the GTP to GDP. So GAP is actually the inhibitor. Here the GAP is called TSC2 or a TSC1 complex. Both the protein actually associate, but the TSC2 is actually very important. they prevent, this is actually gap, this is the gap protein that prevent the REP GTP. Okay, when there is no growth factor, TSC inhibits this REP GTP that actually activate the mTOR C1, so that in the no growth factor, when there is no growth factor, the mTOR C1 is not active, so it is off. You have to understand, mTOR C1 is activated by REP, but REP is inactivated by TSC2. When there is no growth factor, TSC2 inhibit the REP, REP cannot activate the mTOR, so mTOR is off instead. Okay, but when there is growth factor, what happens? I have mentioned the signaling mechanism is, then the Pi3K gets activated, Pi3K activate AKT, or you can say PKB, and then this protein actually phosphorylate. I want to mention here in this red color, they prevent the functioning of TSC2 by phosphorylating this protein. Also TSC1 is present associated with this protein, but TSC2 is important because TSC2 gets phosphorylated. Now this TSC2 no longer inhibit the REP protein. I am here mentioning in red color. It is cannot prevent the inactivation of REP GTP. Now REP GTP activate mTOR C1, and then mTOR C1 can perform its function. I will show you 3 important functions that are performed by mTOR C1. Now the mTOR C1 is on instead. So now we can see how this Pi3K kinase pathway is very related with mTOR pathway. And I have mentioned that this AKTs get activated by mTOR C2 complex that also inhibits this TSC2. What is the full form of TSC2? It is a medical term. It is called tuberous sclerosis complex. Tuberous sclerosis complex, in which benign tumors occurs. Okay, so this is the gap. When gap gets inhibited, then it actually activates. Okay, another thing I want to mention here, that the cell can sense the growth factor by activating the mTOR C1. Also energy, cell can sense the energy. How? When the energy is low, then ATP concentration is low, but AMP concentration gets high, and the ratio when AMP is to ATP ratio is very high, then a protein activated called AMPK, a kinase gets activated. This kinase phosphorylates the TSC2, but by this phosphorylation, this TSC2 gets activated. You can see how much this is different from the phosphorylation by the AKT. When AKT phosphorylates TSC2, it actually becomes inhibited, it becomes off. But when AMPK phosphorylates this TSC2, it becomes on. So the cell can sense the energy inside the cell, so it will actually off the mTOR C1, because mTOR actually helps in the proliferation, growth of the cell. But when there is less of energy, so the mTOR C1 should be inhibited. Now how mTOR actually control this pathway, I will tell you. Now I will show you how mTOR C1 complex actually control cell growth, cell proliferation. The mTOR C1 means it is associated with a protein called Raptor, and this mTOR actually a serine threonine kinase. The same as it is PKB or AKT as a serine threonine kinase, it is also a serine threonine kinase. Now this mTOR can actually perform 3 functions, that is, it helps in the production of ribosome, it produces ribosome. It also helps in the translation initiation or translation promote, it promotes translations, and it actually prevent protein degradation. So this is the main function, promoting the ribosome, translation, increase the translation, actually prevent the protein degradation. How this occur? mTOR C acts as a kinase, so it can phosphorylate. There is a protein called EIF4E binding protein 1. This protein actually capture another protein called EIF4E. This protein is important. This protein actually a cap binding protein. Why it is called cap binding protein? We know that in the mRNA there is a cap like structure. Cap. This protein, when bind with the cap structure of the mRNA, it actually helps in the initiation of the translation. Initiation of translations. So translation actually initiated. This is why it is called eukaryotic initiation factor 4E, that binds the cap and initiate the translation. But EIF4E BP is another protein that captures the EIF4E. This is a binding protein, binds with this protein, so it prevents the function of this protein. When mTOR C actually phosphorylates this protein can no longer bind with this protein, so this protein actually acts as a initiator protein. So by this way it helps in the translations. Another thing, mTOR actually phosphorylates another protein called, or activate a protein called S6 kinase. This S6 kinase can perform two functions. One is it phosphorylates S6, a protein that is present in the small subunit of ribosome, and helps in the increase in translation, because we know the ribosome helps in the translation. So when S6 protein gets phosphorylated, it actually becomes active, it helps in the translations. Another function of S6 kinase is it activates a transcription factor for RNA, Pol1 transcription factor. We know that Pol1 actually helps, the transcription factor called TIF1A. When this transcription factor gets actually activated, it actually helps in the ribosomal or regular ribosomal RNA, small ribosomal RNA. That means rRNA. It helps in the production of rRNA. Okay, ribosomal RNA. And we know that ribosome is made up of protein and rRNA. So it helps in the increase of the ribosome. So ribosome actually produced by this way. This is another pathway. Another thing is this mTOR pathway or mTOR C1 actually perform this function. That is, it actually increase the transcription of some gene or actually mRNA that has pyrimidine in the 5' untranslated region. In the 5' UTR, some RNA has string of pyrimidine. This is called TOP RNA. Why it's called TOP? TOP means tract of pyrimidine. Those RNA at the 5' UTR position has so many pyrimidines. Those are called top RNA. They actually helps in this RNA actually produce ribosomal subunit and also rRNA. That's why it promotes the production of ribosomes. So by this way, mTOR pathway actually control cell growth, cell proliferation and cell survival. I have shown in my for previous class. And insulin can also control the activation of or activity of PKB. So hope this video will help you. Thank you for watching and always keep learning.